This is Dr. Vijaykumar Varma. Today we will be learning on magnetic Janus structures by droplet micromagnetofluidics via this demonstration. Micromagnetofluidics is combination of microfluidics, magnetic fluids, and magnetic fields. So why microfluidics? Because it provides miniaturization of all the operation, magnetic fluids for wireless and programmable operations, and magnetic field to perform all those operation, control all those operation wirelessly. If you use magnetic field generated by electromagnets, then you can program your operations as well. So by this field of magnetofluidics and microfluidics, you can achieve a range of application. For example, magnetophoresis, magnetorheology, ferrohydrodynamics, if particle size is less than 10 nanometer, and of course microfluidics, you can perform micromagnetofluidic and droplet micromagnetofluidic. So micromagnetofluidic is when you don't use droplet, and you can perform many of the mixing and cell sorting applications and so on. You can also combine with optics for detection, optical tweezers, fluorescence activated cell sorting. If you combine in combination with micromagnetofluidics, if you combine electric field, you can achieve electrophoresis and dielectrophoresis. And you can also perform a range of biomedical application for DNA PCR analysis, cytometry, cell analysis. And for chemistry, this is the most promising application of this field. You can perform a range of reaction kinetics, synthesis of Janus particles, chemical analysis, and catalysis. So microcontrol of matter on this microfluidic platform you can achieve via lab on chip, and when you combine with magnetic fluid and magnetic field, all those operations can be wireless. Magnetic droplets, which acts as micro robots, is the most promising application of micromagnetofluidics. You can use a range of configuration, for example, a chip where magnetic structures are deposited, and you can activate those structure to control the droplets motion, which as per your requirement. You can also apply magnetic field for flowing droplets to perform chaining, collection, fusion, and release, and of course merging and mixing of the droplets. And when, let's say, none of these are not your requirements, you can also put some droplets on a surface and then apply magnetic field to achieve its merging and splitting and so on. Hence, magnetic droplets, which acts as micro robots, you can pack anything inside and perform wireless droplet control. Janus structures are very unconventional structures, and they are very non-intuitive as well, because usually whenever we prepare any particles, we we learn to think of, okay, particles can only be in core shell structure or embedded composite like structure and so on. But Janus structures are very interesting, where half of your particles can be one functionality and half of the particle can be another functionality. And you can combine both of them to achieve added advantage of for control, delivery, targeting applications. So the half of the the term Janus came from the Roman god, where half of one face looks towards the future and one face look towards the past, and that's why the month of January. So you can imagine a range of possible uh, a limitless possibility with these structures. So we will be using droplet micromagnetofluidics for this. As the name suggests, it is combination of droplets, magnetics, and microfluidics. So droplets to perform reaction at miniaturized volumes, and you can pack anything inside these droplets. Magnetics to perform wireless manipulation and automation, and microfluidics to perform many such reactions in very short time. The volume range which we deal here is of the order of nanoliter to picoliter. Now you can imagine how many reactions we can perform from one single drop of blood. So if you consider one single drop of blood, volume of one single drop of blood around 1 microliter, you can perform 1000 to 100,000 uh, smaller droplets of the range of nanoliter to picoliter. And if you are able to control all those droplets, you can perform so many reactions. So possibilities of application are limitless for this field of droplet micromagnetofluidics. But today, as the demonstration talk suggests, we will be focusing on Janus material synthesis. Let's start the demonstration of synthesis of magnetic Janus structures by droplet micromagnetofluidic technique. Hello everybody. The field of micromagnetofluidics consists of three parts: microfluidics, magnetic fluids, and magnetic fields to control them on microfluidic platform. Today we will be learning about one of the important application of the field of micromagnetofluidics in the form of Janus particle fabrication. As described earlier, Janus particles consist of two functionalities, and both of the functionalities are directly accessible. So I will be demonstrating a fabrication of magnetic Janus particle, where half of the particle will be magnetic and half of the particle will be having a polymeric phase. The schematic of the setup which I am going to use is shown here. There are two changes. Instead of this flow focusing geometry, I will be using a simple T-junction where continuous phase flows along the horizontal direction and dispersed phase flows along the vertical direction. So this part is not there. And this magnetic field setup which you see here is called as hybrid magnetic field setup, which is combination of uniform magnetic field region and a non-uniform magnetic field region. So that setup I will be using. In addition to that, I will also be using a simple non-uniform magnetic field setup by using a simple single magnet. So our steps which I am going to use, we will be first synthesizing magnetic Janus particles without applying magnetic field. Then we will apply non-uniform magnetic field and synthesize particle. And after that, we will apply hybrid magnetic field and synthesize the magnetic Janus particle. And then we will compare all three results by checking the particles under optical microscope. The platform consists of a high precision syringe pump, UV light source which is shown here, the controller of UV light source where you can control the time, and the microfluidic platform. So this high precision syringe pump is connected to the microfluidic platform. I am using a simple T-junction to produce the droplets. So the droplets are produced by inside the continuous phase of limonene, and dispersed phase is of magnetic phase plus polymeric phase. So the continuous phase is flowing in horizontal direction, and the dispersed phase is flowing in vertical direction, perpendicular to the direction of the continuous phase. Let's take a closer look of the setup. As mentioned earlier, the setup consists of microfluidic platform, magnetic field setup, and the magnetic phase is flowing along this channel. So when droplets are generated in the outlet, which is illuminated by the microscope light, then they travel inside the magnetic field region, and inside magnetic field region, under the effect of magnetic field, magnetic phase gets separated from the polymeric phase, and half of the particle will then attain the magnetic properties and half of the particle will remain non-magnetic. UV light then can be focused by using this microscope lens, and the high precision control of this microscope stage provide the control of the polymerization point. For the synthesis of Janus particle, I will be using continuous phase of limonene, which is shown here, and the dispersed phase, which is enclosed in the aluminum foil. This dispersed phase consists of magnetic fluid plus a PEGDA polymer. A specific composition of PEGDA with photoinitiator was used. So let's start the experiment. The first stage is to fill the syringe with magnetic phase. Which I am doing here. And after filling in the syringe, we need to remove the air bubble inside the syringe, clean it properly to avoid any contamination to the instrument and to other surface while performing the experiment, and mount it on syringe pump. If you have noticed, even though this microfluidic platform utilizes very small volume, I am taking all the precaution like safety goggles, gloves, and so on, because you may handle harmful chemical on this microfluidic platform. So, always put on your PPE, because there is possibility of some splashing of the liquid and so on uh, while performing experiment. Now let's attach the syringe to the disperse phase tube, or the connection to the microfluidic platform, and uh, attach it to the syringe pump. So after attaching, you also need to lock the button on the syringe pump to to enable flowing of that part. So if you see here, I have also put one waste collector. You can also use 2 ml vial for collection of waste, because the flow rate range which we will be using is very small. So this is the limonene phase which I have just showed you, and I have locked both of the syringe, and then uh, entering the diameter of the syringe first, which is 12.5 for this 5 ml syringe, and flow rate of zero point 0.2 ml per hour for disperse phase, and 2 ml per hour for continuous phase. These flow rates I have entered. So once experiments start, then you can wait for some time for the fluids to reach to the T junction and start the and visualize the droplet generation first. Now let's take a closer look of of the droplet generation, which you can see here. The illuminated part is the outlet, and I'm not sure if you can see the droplets very clearly, but I will try to magnify it more so that it will be visible. So droplets are generating in this outlet. There is a very thin line moving along this path. Never mind, you will be able to see it after some time. Let's try again. If you are able to see the droplets in this channel, this is more focused view compared to earlier case. You can see a very thin stream of droplets moving here. However, the resolution is not that good, so you will not able to see it. But when we polymerize the particle after turning on the UV light source, you will be able to see the droplets. See, now it is visible. So I have turned on the UV light source and then adjusted its focus. So it is now brighter. It is exactly focusing on the droplets and polymerizing it in the process. So as long as you keep the UV light on and the flow rate for required for the droplet generation is on, the synthesis will continue. So I am resetting the timer of the UV light. So each time you hear the click, uh, it is resetting the timer of the UV light beam, because if you want continuous synthesis, you can use other UV light source where you don't need that feature. It is one of the safety feature of this UV light. So the process of synthesis is going on now. Let's apply non-uniform magnetic field for the synthesis. So earlier you saw synthesis of magnetic Janus particles. They are actually not Janus because there was no magnetic field applied. So composite magnetic particle you can call them. So now let's apply non-uniform magnetic field, which you see here. by using a permanent magnet. So I am adjusting position of the magnet little bit. This is one of the, you can say, tricky situation with the permanent magnet. You need to place the magnet very carefully. And the position of the permanent magnet need to be very precise. I'm not doing it very in very precise manner, but still uh, the position which a position which will be good enough for this experiment. Now let's perform synthesis uh, with this non-uniform magnetic field. So magnet is located here, and now synthesis is going on. You can clearly see the stream of droplets moving here and then getting polymerized. Thus, the bright light which you see here is due to fluorescence of the dye I am using inside the polymeric phase. So this synthesis will go on for some time, and when we collect enough amount of this magnetic Janus particle, these are not these are not precisely Janus structure, but still some magnetic content will get sorted and show a specific configuration, which we will see by the end of this demonstration. Now let's apply the hybrid magnetic field, which is this specific setup of two magnets, which I showed earlier in my schematic. So I am placing the outlet channel between this combination of magnets. So this specific combination of magnets allows or provide a non-uniform magnetic field to pull particles and uniform magnetic field to align particles. Let's begin the synthesis at hybrid magnetic field, which is combination of uniform and non-uniform magnetic field. You can see droplets are moving and UV light is applied. You can't see it clearly because it is between these two magnets. So exactly similar to our earlier experiments. We will be continuing this experiment till sufficient amount of particles are getting collected. Cleaning and washing of magnetic particle um, is, you can say, one of the most efficient step. You can just apply magnetic field to sort out particles and perform washing repeatedly. So that is also one of the main advantage of using magnetic beads. because you can apply magnetic field and then separate it from the content. So I'm performing magnetic washing of these particles. So pouring some DI water and washing it. Let's look closely towards magnetic washing. So I'm taking the magnet, shaking the solution with magnetic particles and bringing it close to the magnet and removing the waste from the container. And after washing, I'm transferring the solution to the petri dish for observation under optical microscope. We used optical microscope to compare results. BF represent bright field mode and DF represent dark field mode, which correspond to fluorescent response of the particle or fluorescence of the particles. The first column correspond to particle synthesis under no magnetic field. Second column corresponds to particle synthesis with non-uniform magnetic field, and the last column corresponds to magnetic particle synthesis or Janus particle synthesis under hybrid magnetic field. Evidently, the first rule, which correspond to synthesis without magnetic field, shows particle dispersion very uniformly. So magnetic particles are dispersed throughout the particle, and there is uh, no change in fluorescence. So it is also uniformly distributed throughout the particle. The second column, which correspond to particle synthesis under non-uniform magnetic field, shows of band formation of the particles under which is due to non-uniform magnetic field. However, these bands are distributed throughout the particles, so you can say it is partially in Janus configuration, and also the fluorescence response where particles are present is not there, and the remaining portion where magnetic particles are not there, there is fluorescence response. The last case, which correspond to hybrid magnetic field, shows the Janus configuration where magnetic particles are nicely distributed on or separated on one side of the particle, and the same side does not uh, show the fluorescence under dark field mode. Hence, in summary, we develop non-uniform and hybrid field-based microfluidic platform which utilizes droplet micromagnetofluidics for the synthesis of Janus particles. We demonstrated synthesis under different magnetic field conditions and compared without magnetic field condition. There are different types of dominating factors, so which we did not investigate here, but in general they present in any of the droplet micromagnetofluidic setup that we realized after a very detailed investigation which we performed under that specific project. So the dominating factors which are very important for DMF setup are flow rate ratio, magnetic field gradient, magnetic field strength, interfacial tension between the continuous and disperse phase, concentration of magnetic nanoparticles, and all this, if you are able to tune properly, then you can achieve a superior programmable control. Other factors which are also very important are viscosity, field gradient, and MNP concentration. So these are also applicable for micro-magnetofluidic setup. This ends the demonstration. Thank you for your attention. Now I am open for the questions.